so today's lecture is on cellular respiration. Now, in the past, we've talked about processes that are anabolic and catabolic, right? So cellular respiration, we'll look at this in a second, is actually a catabolic process, the process of breaking down large complex molecules and extracting free energy from them. So what cellular respiration does, it'll actually take a glucose molecule, which is a molecule of relatively high free energy, and break it down into carbon dioxide and water, and extract that energy and transfer it to molecules of adenosine triphosphate, or ATP. Now remember, this is just a rule of thumb, and this is review from freshman year, that aerobic respiration is a process that uses oxygen. Anything that is not aerobic or deemed anaerobic does not use oxygen. And there's two forms of anaerobic processes that we'll talk about, one being anaerobic fermentation and the other being anaerobic respiration. So this process occurs, and we know this, it occurs in the mitochondria. We've talked about this over and over again. The mitochondria is the powerhouse of the cell. You know, I hate that term because it's lame. But remember, this is why we call it that, because it makes a majority of the ATP. It's just a funny meme. So in order to understand cellular respiration, we first have to understand where cellular respiration occurs. So the mitochondria actually has a double membrane. It has one surrounding the outside and has an inner membrane, which we'll call the cristae. Those cristae are the folds of the mitochondria. Now, thinking back to what we know about folds in biology, as we've talked about them so far, the goal of those folds is to increase the surface area for something. In this case, we're increasing the surface area to have more area for reactions to occur, for cellular respiration, specifically the electron transport chain to occur. All right, and we'll talk about the electron transport chain later. But another cool thing to notice is that the mitochondria, it has a double membrane. So why does it have that? Remember, we've talked about this previously, that the mitochondria is hypothesized to have once been its own free-living organism, a prokaryote specifically, that was engulfed by an ancestral eukaryote, forming a double membrane structure. That bacteria, or that prokaryote, was very efficient at doing cellular respiration. That eukaryote was much larger. It was efficient at getting food for them. And as a result, they formed a symbiotic relationship. And the evidence that we have for that is the double membrane that we just mentioned, but also the circular DNA that is within the mitochondria that you can see right in this area, right here, and the green ribosomes that are floating. So it has its own ribosomes and its own DNA, which is really neat. All right, so the famous reaction. Carbon dioxide and water are formed when you break down glucose using oxygen. This is very similar to what happens when you use gasoline, or when you burn basically anything, when you burn a carbohydrate or you burn a fat or a lipid. When you, when you burn it, oxygen is required for that process to happen. So you take your C6H12O6, you add oxygen, you break that down into carbon dioxide, water, and ATP energy. The same thing happens in your car. You put fuel in your car, the fuel gets pumped and aerosolized using a fuel injector, and oxygen is mixed with it, and you ignite it with a spark plug, and what comes out? Carbon dioxide, water, some carbon monoxide in that process as well, and mechanical and heat energy. Our body does the exact same thing. It just does it in a more efficient way. And we transfer a lot of that energy, instead of to heat, which we do some of that, to ATP molecules. And the way you can remember the inputs and the outputs of this reaction is that what do you take in? You take in food or sugar. You take in oxygen. And what do you breathe out? You breathe out carbon dioxide. And if you've ever been in a car when you're a kid during the winter, if you blow on the window, you form water. You breathe out water vapor, and it condenses on the window itself, and you draw something. So that's a quick and easy way to remember just the basic inputs and outputs of the reaction. Now, you're going to have to know it way more in depth than that, so this is going to be like a drop in the bucket compared to what you have to know, but it's just good to know that specific mechanism for remembering it. All right, so remember, everything that we're talking about in this process is made possible by enzymes, the things we just talked about in our last unit. Remember, they're made of proteins. They have a specific structure that is determined by the order of the amino acids. That's the primary structure. You can see a bunch of, if you look in the actual protein itself, you can see a bunch of alpha helices and some beta sheets up here. That's showing its secondary structure. How it starts to fold is its tertiary structure, and then putting all the subunits together would be the quaternary structure. Okay, but these enzymes are driving this process. It's speeding up the chemical reactions by lowering their activation energy. So another thing that you want to know before we really start getting into the process is this idea of oxidation and reduction reactions, or redox reactions, which you learned about during your sophomore year in chemistry, or if you took AP chemistry last year. So all redox reactions are the transfer of electrons from one reactant to another one. And the thing that loses electrons is the thing that gets oxidized, and the thing that gains the electrons is reduced. Well, why is it called reduction? It's because you're adding negative charges, and when you add negative charges to something, you reduce its overall charge, or its oxidation state. So it's called reduction because you're adding more negative charges to it. The simple way that you probably were taught to remember it was Leo goes Gur, lose electrons oxidation, gain electrons reduction, but this is a more, I think, satisfying explanation, which is when you add negative charges to something, you're reducing its charge. So another cool thing that you probably didn't learn about, because it's not, in regular chemistry this isn't the rule, but in organic chemistry it tends to be, is that anytime you oxidize something and you remove electrons from it, you're actually going to reduce, or you're actually going to make the charge more positive. And when you gain electrons, you make the charge of that thing more negative. But you have to have a neutral product in the end usually. So what usually follows the electrons are hydrogens. So you can tell the thing that gains hydrogens is the thing that has been reduced, and the thing that has lost hydrogens in organic chemistry is the thing that has been oxidized. Now if you look here at glucose C6H12O6, it gets oxidized to form CO2. Notice what it lost. It lost its hydrogens. Same thing here. If you look at the oxygen gets reduced in this process. It gains electrons, and we're going to see through this process how that actually happens. But how can we tell that it has been reduced? Because it has gained hydrogens. So that is the simple way you can tell if something has been oxidized or something has been reduced. All right, so let's begin talking about the actual process. So aerobic cellular respiration requires oxygen at the end. It's actually going to be what we call our terminal electron acceptor. Now, that doesn't mean anything for you right now, but it will be once we get there. This extracts tons of ATP per glucose molecule compared to anaerobic types of anaerobic fermentation. So you get 36 to 38 theoretical ATP, but we don't actually get that many in reality. And we'll kind of count them and tally them as we go through the process, okay? So cellular respiration has 3 steps you have to understand: glycolysis, the citric acid cycle, or the Krebs cycle, and the electron transport chain and oxidative phosphorylation. And the overall purpose of this is to take glucose, rip it apart, and extract as much energy as we can from that exergonic process. Now, in order to do this, we are going to create some ATP initially, okay? But the goal is to create more ATP later by creating these things called electron carriers. And there's 2 types, which we're going to go over. But think of electrons as being high energy particles. The flow of electrons is electricity that can be used to do work. The same thing's true kind of in here. When you add electrons to things, they become more high energy. So our goal is to take these electron carriers, transfer electrons from glucose to generate ATP. All right, so here's the overall process looking at all 3 phases. You can see here we have glycolysis right there, we have the citric acid cycle right here, and we have oxidative phosphorylation, which includes 2 processes called electron transport and chemiosmosis, and that is the 3rd and final step, which generates the majority of the ATP compared to the other 2 if you look at them. Now, notice that initially we create a little bit of ATP down here. 2 and 2, 4 total ATP, not very much. But what we get out of glycolysis in the citric acid cycle are all of these electron carriers, NADH and FADH2. Those are going to be used, as you can see, they're going into this last process to generate a majority of the ATP. Now notice I have a lower number than I said earlier here, 32. That's because this is that's closer to the actual value, not the theoretical one of how many ATP are generated. All right, so let's talk about glycolysis. First, what does that mean? Glyco means sugar, lysis means to split. All we're going to do in this first step is we're going to take sugar and split it in half. So if glucose has 6 carbons on it, C6H12O6, we're going to create two 3-carbon molecules out of it called pyruvate. Okay? Now glycolysis is universal in all organisms, and it's the only one of the steps that we're going to talk about that does not occur in the mitochondria. It occurs in the cytoplasm of the cell, and you have to remember that. Okay? But every single organism that exists does glycolysis, meaning that it likely, or it did, evolve very early on in the evolutionary process. Okay? And as I said, we're just taking glucose, we're splitting it into two 3-carbon molecules, and all we're going to get out of this is a little bit of ATP, 2 in fact, but a bunch of electron carriers that we're going to use later called NADH. Here's showing you the summary of the process. Here's our 6-carbon glucose molecule. Notice what we get at the end. We just split it in half into two 3-carbon molecules called pyruvate. What's kind of interesting here though, like notice the inputs. Some ATP has to go in. It's kind of like the activation energy of the process. Now this is a process that requires lots and lots of steps. It's not just one reaction, it's several reactions, as we'll see in a minute. But we have to put a little bit of ATP in to get a little bit more out. So we put 2 in, we get 4 out, we get a net of 2 ATP. What we also put in are these NAD pluses, and these are going to rip electrons off of glucose and transfer them onto a molecule called NADH. Now, how do I know which is the oxidized version, which is the reduced version? Remember what I said before. If you add hydrogens to something, that's usually because they followed electrons. So if this is NAD+, we add 2 electrons to it, one to neutralize the making it NAD, another one to make it NAD-. What follows, an H+ molecule to neutralize it, we get our NADH. So our products at the end of this whole thing are 2 pyruvates, we get 2 net ATP, and 2 NADHs, which are electron carriers. This actually looks at the entire process of glycolysis, more complicated. Now in college, you're going to have to memorize every single step of this, every single name of every single molecule, the structure of the molecule, and this is even oversimplifying it a little bit because it doesn't show the actual structures, it just shows the number of carbons. But I'm going to point out some interesting things here that will actually help us review some of the things we've talked about previously. So if you look at the top, you see the molecule glucose going in right there, and notice the first thing that happens is our first ATP goes in and donates a phosphate group. So the first question is, what do you think the name of that, the general name of the type of enzyme that first process is driven by? It's a kinase. Why? Because it took a phosphate group off of ATP and transferred it to a molecule. Okay, you're going to see that again. There's another one here. We take an ATP and we add another phosphate group, and it's called fructose 1,6-bisphosphate. Bi meaning 2 phosphate groups, says it now has 2 phosphates on it. And if you actually look at the enzyme that drives this, it's called phosphofructokinase. It's a kinase, and this is the actual enzyme. It takes an ATP, which was put in the active site, transfers it to this molecule right here. So that's the actual process, and it shows the substrates binding to the active site. But what's really cool about this, and it's a review of what we talked about before, there's a regulatory site on the other side of it. And there's there's more than 2 molecules, but 2 I'm going to focus on that will bind there and either speed up the reaction, causing the enzyme to be turned on and work more efficiently, or it'll slow it down. So if ATP binds, think about this. This is an example of negative feedback or feedback inhibition. If we have enough ATP, we don't have to really make more of it. So ATP will bind to that regulatory site, slowing down substantially the speed of this reaction, basically changing the shape of the enzyme. If AMP, which is adenosine monophosphate, which is a much lower energy molecule, is present, then that will bind to the regulatory site, and it will cause it to speed up. It'll actually turn the enzyme and cause it to go faster. So it allows for control. If we have a lot of ATP, we don't have to do this process as much. If we don't have a lot, because we have AMP, remember that's the less energetic form of ATP, where there's AMP, ADP, and ATP, with ADP kind of being in the middle. If there's AMP present, then we want to make more energy, so it's going to cause this to work faster. It's just a great example of negative feedback. But notice, we put our 2 ATP in, we get a total of 4 out, and what do we get at the end? 2 pyruvate molecules and 2 molecules of electron carriers, NADH. This is showing you what I meant previously about how ATP inhibits phosphofructokinase. Citrate, which is one of the products of the citric acid cycle, will also do that. You don't really have to worry about that as much. But AMP stimulates it, because AMP is a lower energy molecule compared to ATP. And if we have more lower energy molecules, that means we have to make more higher energy ones. Now, glycolysis creates ATP through something called substrate level phosphorylation. And this is very similar to what happens with kinases, except the opposite. So in substrate level phosphorylation, instead of transferring a phosphate group from ATP to the substrate that's in the enzyme, we're actually going to take a phosphate group, transfer it from a substrate back to ATP. That's why it's called substrate level phosphorylation. Now, only a few ATP per glucose molecule are made through this mechanism. Most are made through something called oxidative phosphorylation, which is a completely different mechanism for making ATP, which we'll talk about later, that is way more efficient. All right, so here's the first question. Glucose is made from 6 radioactively labeled carbon atoms. It is fed to yeast cells in the absence of oxygen. So there's no oxygen in this case. And usually you can only do glycolysis in the absence of oxygen, which we'll talk about. How many molecules of radioactive pyruvate are formed from each molecule of glucose? So they're just saying from one molecule of glucose, which has 6 carbons, how many pyruvate are formed? This should be pretty easy. The answer is 2. Because you take the 6, you can divide it into 2 3-carbon pyruvates. All right, so now what? We have a couple ATP, we have a few electron carriers, and we have this stuff called pyruvate. Well, our goal now is to actually completely break down that pyruvate and remove all of the, not all of it, but most of the free energy from the rest of the molecules. So in order to do this, we have to now shuttle that pyruvate into the mitochondria. So now we're getting into the mitochondria's role in all of this. Now what this is going to do overall, just it's super simple, it's going to pluck off carbons one by one. And as it does it, it's going to turn those into carbon dioxide, the stuff that we breathe out, okay? And in the process, we'll create a little ATP, but more importantly, we're going to create more electron carriers. Now we're going to create more NADH, but we're also going to create a new one called FADH2. Now, that sounds confusing. Remember, they're just these molecules that hold electrons, and we're going to transfer them so we can use the energy from those electrons later to generate lots of ATP. Okay. So there's 2 steps that we kind of focus on with the next part of this. There's the link reaction, and then the Krebs citric acid cycle. The link reaction, the goal of that is to just rip off 1 carbon molecule from pyruvate. So if pyruvate has 3 molecules of carbon, and we rip off 1, how many carbons will be left? 2 carbons, all right? And in the process of doing that, we create carbon dioxide. That's where the 1 carbon goes. And we also create and transfer electrons to 1 molecule of NADH, which is an electron carrier, okay? The electrons go on NAD+, electrons, and form the reduced form, which is NADH. We then take that 2 carbon molecule that results, right? So we take, as we said, we start with a 3, we go to a 2, and then we take that 2 carbon molecule and we're going to throw it into the citric acid cycle. It's going to attach a coenzyme called coenzyme A to this acetyl group, and the acetyl group has 2 carbons, allowing it to react more efficiently, and in doing so it's going to fully oxidize the remaining 2 carbon molecule. And in doing so, it creates 1 ATP, 3 NADHs, and 1 FADH2, releasing 2 carbon dioxide because there are 2 carbons left. Okay, now that's per the initial pyruvate molecule. So remember, if we put 2 in, we get then 2 carbon dioxides out, because if you have 2 3-carbon molecules and remove a carbon dioxide from each, you end up with two carbon molecules. And then all of the stuff that's written about the Krebs citric acid cycle below is per acetyl-CoA, or the 2 carbon molecule. So we actually would end up getting 2 ATP, 6 NADHs, 2 FADH2s, and then 4 carbon dioxides out of that process. So here's showing you that link reaction. Now this happens in the matrix of the mitochondria, which is this inner part that I'm kind of highlighting up here. Okay, that's just part of it. All of that where it looks like there's water is considered the matrix. And what we get out of this is 2 NADHs for the 2 pyruvate, 2 acetyl-CoAs, and 2 carbon dioxide gas molecules. And here's now talking about the citric acid cycle. Again, this also occurs in the matrix of the mitochondria. This rips off the last carbon dioxides, creating an FADH2 and 3 NADHs per acetyl-CoA. And as I said, per glucose, initially in the Krebs citric acid cycle, you'll get 2 ATP, 2 FADH2s, and 6 NADHs. Now remember, what are those? They're just electron carriers. Don't get bogged down with how all these letters and things. The goal is to generate some ATP, which we get, but it's also to create these electron carriers, which we're going to use later. And then what we'll do is we'll remove all of the carbon dioxide, all the carbons that were initially in the glucose as carbon dioxide gas. And this is showing you, again, the actual Krebs citric acid cycle with their structures. In college, you'll be required to know the name of each one, the enzyme that drives each one of those arrows, or each one of those reactions. But in this course, you don't have to know it at this level. You just want to know what is going, or at that level, I should say. What are the inputs, what are the outputs, and what are the important things we're extracting from each process. So in this case, ATP is still generated by substrate level phosphorylation, the transfer of a phosphate group from a molecule back to an ADP molecule making ATP. So what have we made so far? We've gotten 4 ATP, 2 from glycolysis and 2 from the citric acid cycle. We've created 10 NADHs, 2 from glycolysis, 2 from the link reaction, and 6 from the citric acid cycle, and 2 FADH2s that we got from the citric acid cycle. So notice we've created a lot more electron carriers than we actually have created ATP. So now we're going to talk about the importance of these electron carriers in generating a majority of the ATP that we're going to get through the last process. So these electron carriers, this is their structure. Now if we look at them, you should be able to figure out what overall type of macromolecules are these. We have phosphates, we have sugar, we have a base, we have a sugar, we have a base, we have, again, this is the FAD molecule. We have sugar, base, we have phosphate groups. This is a nitrogenous base right here. This is actually a kind of a sugar molecule. Notice the ratio of carbon to hydrogen to oxygen is 1 to 2 to 1, or I should say a sugar component to that molecule. So these are nucleic acids. That's what they are. They just have a different type of role than the other nucleic acids we've talked about. Like ATP is another type of nucleic acid. DNA and RNA are types of nucleic acids. So you'll notice this is the spot that changes in the NAD plus versus the NADH, where there is electrons versus not electrons. Okay, the same thing is true here. The difference, the reduced form is right in this region. Notice it gained an H off that nitrogen, and electrons followed it, or the H's followed the electrons to it. So this is just showing you some kind of like review about the structures, but also like you don't have to know the structures of NADH and FADH2, but recognizing them as nucleic acids is something you should be able to do. All right, so here is the fun part now, the part where we're going to get lots and lots of ATP using those electron carriers we were just talking about. So basically, what is going to happen in the process of electron transport and chemiosmosis, collectively called oxidative phosphorylation, is that we're going to transfer the electrons from the carriers to several different protein complexes in the mitochondrial inner membrane. And as they're passed, they're going to pump hydrogens across the membrane, and those hydrogens are then going to flow back across the membrane, almost like water flowing through a hydroelectric power plant, and it's going to cause a protein to spin, and that protein is going to generate lots and lots of ATP. So here's showing you that process. This is actually the electron transport chain. So right here we have our mitochondrial inner membrane, and we have 4 different protein complexes, 1, 2, 3, and 4. And then we also have several shuttle proteins called cytochromes. This is coenzyme Q10, this is cytochrome C. You don't have to really know their names, but basically what these cytochromes do is they're going to act as shuttles to help move electrons from complex to complex. So where do the electrons come from? They come from those electron carriers. So if we look down here, we have our NADH. Our NADH drops off electrons, and electrons are passed to complex 1. They are then going to go from complex 1 to coenzyme Q, to complex 3, to cytochrome C, eventually to complex 4, with their final resting place being on water. How does it become water? Well, the electrons end up on oxygen. So the electrons are passed to the oxygen, hydrogens follow them, and they become molecules of water. So why did we skip complex 2? What's the point of it? So FADH will go and pass electrons from complex 2 to coenzyme Q to 3 to 4 and eventually onto water. Now there's a difference there that we're going to talk about once you understand the purpose of actually passing these electrons from complex to complex. So the reason we do that is the potential energy of them passing from the oxidation and reduction of each of these individual protein complexes. We use the energy to pump hydrogens across against the concentration gradients. This is kind of like a form of active transport, similar to when we talked about co-transport, right? So these hydrogens are getting pumped across, but not using ATP. They're getting pumped across using the potential energy of these electrons passing. This is creating a high concentration of hydrogen ions in the inner membrane space. So up there where it's more orange is the inner membrane space. Down here where it's a little bit lighter, like a yellowy color, is the mitochondrial matrix. Now, what will that do to the pH up in the matrix versus in the inner membrane space? Well, if we're adding hydrogen ions to the inner membrane space, that should lower the pH. What are you going to do as you pump hydrogens out of the matrix? You're going to raise the pH of the matrix. Okay, that was just a quick review of acids and bases. Now notice, only complex 1, 3, and 4 pump hydrogens across. Complex 2 does not. Okay, and that's going to come into play in a little bit. So what's the purpose of these hydrogens being pumped across? So those hydrogens are then going to go, and they're going to flow back through this very special and important protein called ATP synthase. It's basically like a jet engine that spins as hydrogens flow down through it. Almost, as I said, like a hydroelectric power plant. And in doing so, it's going to force molecules of ADP and inorganic phosphate together and generate ATP at a very fast rate. So through this process, we end up generating about 32 approximately, depending on how you look at it, but theoretically, about 32 ATP. Just through this process. Now notice, before, just through glycolysis and citric acid cycle, we only created 4 previously. So what are the differences between the words chemiosmosis, electron transport chain, and oxidative phosphorylation? So collectively, the electron transport chain, which is the passing of the electrons and pumping hydrogens across, and the process of chemiosmosis, which is the hydrogens flowing back across the membrane, causing ATP synthase to spin, the whole collective thing is called oxidative phosphorylation. And as I said, remember, this generates lots and lots of ATP. So as I said before, the protein complexes will pass electrons to cytochromes, which are these movable electron carriers, and they're passed from complex to complex, and as they do that, the complexes pump hydrogens across. That last thing that accepts the electrons at the end, after complex 4, is oxygen. So oxygen is the terminal electron acceptor. It is the final thing that is reduced. And when things are reduced, hydrogens go with it. So oxygen and hydrogen form water. As I said, they're going to flow back across the membrane through the ATP synthase molecule. That's the only place that they can get across because remember, hydrogen ions are charged and they cannot easily cross the membrane. They have to go through some place that has an environment that is favorable for them to cross. And as they cross, they're going to cause ATP synthase to spin, and ATP is going to be generated very quickly. So this is showing you again the electron transport chain, which is this process right here. And the process of the hydrogens flowing back across is chemiosmosis. And it's this process that actually generates the ATP. This is looking at the ATP synthase molecule. Now it is a very complicated protein. These are all different subunits all put together to form this complex protein. So this is showing quaternary structure of ATP synthase. If I were to ask you about the amino acids that are located on the inner part of the membrane, remember you would probably assume that the R groups on those have mostly nonpolar amino acids on them. The same thing's true with this A subunit over here. Whereas there may be more charged subunits in the alpha and betas and sigmas and things like that in the F1 part of the complex. But again, the whole role of this protein is to spin and generate ATP as hydrogen's ions flow through it. It's kind of like this. This is the, I did not draw this, but the simplified idea here. You have ADP coming in, and the ADP plus the inorganic phosphate is being generated through the flow of hydrogen ions down the concentration gradient. Similar to how grain used to be ground in a mill in the early days before we had electricity. All right, question. When electrons flow along the electron transport chains of the mitochondria, which of the following changes occur? All right, so let's check and think about this for a second. Okay, so the first thing we're going to check is the pH of the matrix will increase. Is that true as electrons flow across the electron transport chain? Let's look back to it. Whoops, went too far. We can use this picture. Notice we're pumping hydrogens out of the matrix and into the inner membrane space. What's that doing to the inner membrane space? It's lowering the pH. What's that doing to the matrix? It is raising the pH. So that answer makes sense. So far, so good. We have an answer to that. But let's check the other ones real quick. The ATP synthase pumps protons by active transport. That is not what the ATP synthase does. Hydrogen ions or protons flow through it with the concentration gradient and causing it to spin, generating ATP. The electrons don't gain free energy during this process. They actually lose free energy as they're passed, and that free energy is transferred to the process of hydrogens flowing across the membrane. The cytochromes of the chain phosphorylate ADP to form ATP. No, that happens in ATP synthase. NAD plus is oxidized. No, NADH is oxidized in this process because the electrons are being removed from it. Okay? In the presence of metabolic poison that specifically and completely inhibits the function of mitochondrial ATP synthase, which of the following would you expect? So you inhibit ATP synthase. Now think about what ATP synthase does to answer this question. It generates ATP, right? So if you inhibit that, you're not going to make much ATP. But also it's going to stop the flow of hydrogen ions back across the membrane. Okay? So let's check each one of these. A decrease in the pH difference across the inner mitochondrial membrane. It would actually cause a bigger increase because there's no flow of the hydrogens back across it. So it can't be A. An increase in the pH difference across the inner mitochondrial membrane. That could be true. So let's hold off on B. That's a good answer because you're constantly pumping the hydrogens across, but they can't flow back to kind of reach the equilibrium in what they were before. Increased synthesis of ATP? No, it would decrease it because it doesn't work. Oxygen consumption would cease. The oxygen consumption wouldn't cease. You keep consuming oxygen. You would just keep pumping hydrogens across, and the oxygen actually wouldn't do anything. You wouldn't feel like you're being satisfied by breathing the oxygen in. And then the last part, the proton pumping by the electron transport chain would cease. No, it would keep going, as I said, but it would just increase the pH differences between the matrix and the inner membrane space. So the answer to this one is B. Cyanide, famous poison, right, in history that a lot of, like it was commonly used in World War II by the Nazis to get cyanide pills that they got caught, they would just bite into it and it would kill them. So why does that happen? Why is cyanide so dangerous? So it's a poison that will actually block the passage of electrons along the electron transport chain. Which of the following is a metabolic effect of this poison? So if you can't pass electrons, you cannot pump hydrogens, and if you can't pump hydrogens, you make no ATP, and if you make no ATP, you die very quickly. So the lower pH in the inner membrane space is much lower than normal. No, because we're not pumping hydrogens up there. That would keep it less lower than normal, so it can't be A. I know these all say A. I'm sorry. Let me change them. A, B, C, D, and E. Electrons are passed directly to oxygen, causing the cells to explode. That makes absolutely no sense. Alcohol would build up in the cells. The problem with this is humans actually, and we'll talk about this, humans do not create alcohol through our anaerobic processes, so that can't be right. It's only like yeast and bacteria that do that. NADH supplies would be exhausted and ATP synthesis would cease. You would stop, NADH supplies would not be exhausted because you cannot pass the electrons from place to place. Now let me actually point out and explain this for a second. Let me go back what I mean by that. Okay, so why do we die when we don't have oxygen? So if oxygen does not take the electrons forming water, the electrons will back up. They'll back up on complex 4, on cytochrome C, on complex 3, and coenzyme Q, and complex 1. They'll back up all the way. So at that point, because there's already electrons on complex 1, well, it's not going to want to take electrons from NADH. So the NADH isn't going to lose its electrons. So no oxygen present, the whole process backs up. No hydrogens pump across, and if no hydrogens pump across, we can't generate ATP. So that's why having oxygen is so important. Otherwise, we start generating oxygen through anaerobic fermentation in our muscles, or in any of our cells. So NADH supplies would not be exhausted, as I just explained. ATP synthesis would still cease, but the NADH supplies wouldn't be exhausted. No proton gradient would be produced and ATP synthesis would cease. That is the best answer, A. That makes sense because you don't actually make a proton gradient because you're not pumping hydrogens across. And if there's no hydrogens to pump across, there's nothing to flow across the membrane through ATP synthase to generate ATP. One other thing I want to point out too is that FADH2 down here actually generates less ATP per molecule than NADH does. And why do you think that is based on what we talked about? Take a look at it. So if hydrogens are the currency that is used to create ATP, notice FADH2 only pumps at complex 3 and complex 4, whereas NADH pumps at complex 1, 3, and 4. So because that's the case, FADH2 actually generates less ATP per molecule than NADH does. All right, so here's an overview of the process, right? We have our where does our glucose initially go in? It goes in at glycolysis, okay? Where does our oxygen initially come into play? Our oxygen goes into the electron transport chain. That's the terminal electron acceptor. Where are carbon dioxides released? From the link reaction in the citric acid cycle. Where is water produced? As a result of the electrons being passed onto the oxygen at the end of the electron transport chain. And what do we get at the end? A whole bunch of ATP, with most of it coming from the process of oxidative phosphorylation. Okay, so what if there's no oxygen at all, or very little oxygen? What does our cell start to do? A process called fermentation. It's called anaerobic fermentation because that's when there's no oxygen present. So if I were to ask you, what do you remember from living environment? The common thing that people would say is, well, there's no oxygen, and you get a little ATP, and you generate byproducts, such as lactic acid and alcohol. You did a lab where you pinched clothespins over and over again to get lactic acid to build up in your hands, and you talked about that process a little bit there, but it was mostly like an experimental design type of lab that you did. But anyway, what you had to know is load a little oxygen, little ATP generated from it, so aerobic is much more efficient, but you also get these byproducts which can build up and cause problems. So, but why, like, how does this happen? This process is essentially just glycolysis, so you get 2 ATP, very little ATP, but it's an added step where we have to regenerate our NAD+, because if you're doing glycolysis constantly, the reactants of that process are the glucose, some ATP, and NAD+. Those go in. The products are NADH. Now, what you know about Le Chatelier's principle that we talked about before is that if the products start to build up and we don't regenerate the reactants, then we're going to kind of slow down this whole process. So we have to regenerate the NAD+ somehow. Okay? So what we do is we generate ATP through glycolysis. There it is, our 2 ATP. And we also generate our NADHs. But if we want to keep doing anaerobic fermentation, we have to regenerate our NAD+, because that's one of our reactants. So what we'll do is we'll actually take the NADHs and we'll take the pyruvate that was a result from glycolysis, remove some carbon dioxide, which is what is the carbonation that's found in some forms of alcohol like beer. It's converted to acetaldehyde, and then finally it removes the electrons from NADH and converts it into ethanol, which is alcohol, which is a byproduct. Now in human beings that would be a big problem if we generated that byproduct, because it would cause our nervous systems to be inhibited and slowed down. So we generate instead lactic acid as opposed to alcohol. So the pyruvate are converted into lactic acid. Remember, the goal of it, though, is to regenerate our reactant NAD+ from the NADH. Now, in normal aerobic respiration, the NADH is converted back into NAD+ when it donates its electrons to the electron transport chain. But that doesn't happen here because we have no oxygen to take the electrons away from the final complex, and thus normally, if we didn't create these byproducts, lactate would build up in our muscles, and that's when you get, like, if you're doing a lot of anaerobic fermentation in your muscles, you get cramping and like muscle soreness and things like that, which limits the amount of reps you can do if you're lifting weights and the amount of running you can do if you're a runner. So speaking of ethyl alcohol, this is really cool. When it's consumed, the way it actually works, why does it cause inebriation? Why does it cause your nervous system to slow down? It'll actually attach to a special receptor called a GABA-A receptor, and it as an enhancer of GABA. So what's weird about GABA is actually inhibits stimulatory signals. So if GABA inhibits stimulatory signals, when it's activated, it actually causes the nervous system to slow down. So it doesn't react as fast as you want it to. And that's an important control mechanism for normal functions in the body. But because ethanol actually increases the binding of GABA to GABA receptors, where GABA is a neurotransmitter in your nervous system, you'll have an increased amount of inhibition, less stimulation, and thus you feel inebriated, or you have a depression of your nervous system. It slows down, and that slows down your reaction time and causes all the effects that alcohol has, until finally your liver will process it appropriately and break it down the way it needs to. So here's showing you the GABA receptor right here. Notice the membrane. Okay, this is an example of an ion, ligand-gated ion channel, where GABA, the neurotransmitter, binds. It's the ligand. It causes this to open, but when ethanol binds to this thing, it actually enhances the ability for GABA to react, therefore causing GABA to work more often. And because GABA inhibits processes, it increases the amount of inhibition that GABA has, therefore causing you to feel inebriated. Another cool thing is alcohol is also a diuretic. That means it causes urine output to increase. So when you consume lots and lots of alcohol, you tend to urinate more frequently. And that's because diuretics stop aquaporins from binding to your kidney cells. That thus, if your kidney cells can't reabsorb water from your urine, the water is going to stay in your urine. It's actually an important mechanism for maintaining water balance in your body. And if you cannot reabsorb the urine, you're just going to urinate it out, all that water, and thus you'll become dehydrated much quicker. So it's so important that that you stay hydrated, especially if you take any diuretic. There's some things called water pills, which are also diuretics as well. Um, Last thing, there's also anaerobic respiration, which is not anaerobic fermentation. It's a different thing technically. Sometimes other teachers or even they use these words, anaerobic respiration and fermentation, interchangeably. They are not. They are different things. So it's more similar to anaerobic respiration than to fermentation, actually, but instead of using oxygen as a terminal electron acceptor, it uses things like sulfates and nitrites, other types of chemicals, to be the terminal electron acceptors that take the electrons away from the electron transport chain, but it would still use chemiosmosis, it would still use ATP synthase, but it allows them to generate lots of ATP even though they're in anoxic or low oxygen environments. This doesn't happen in humans, it happens in extremophile bacteria and things like that that live in extreme parts of the planet. So, and as I think I mentioned this previously, we saw this too, but glycolysis, we're pretty sure that it evolved very early on. And the common ancestor of all life on Earth actually did glycolysis. So all living things do this process. It is fundamental. And technically, all organisms did it until about 2.4 billion years ago when oxygen first showed up. So oxygen wasn't around. There was no oxygen in the environment at all. And oxygen, when it first started showing up through the result of photosynthesizing organisms, because photosynthesis must have evolved first before cellular respiration, because cellular respiration needed oxygen to happen. Photosynthesis actually killed lots and lots. There's a huge die-off because oxygen is a destructive molecule. It rips electrons off of things. It's an oxidizer. That's where that name comes from. So, so organisms, only the ones that were adapted to be able to deal with oxygen, were actually able to do so and survive. We mentioned this already, the endosymbiotic theory, but I'm going to talk about it one more time because it's important to understand. Remember, there's tons of evidence that we have that mitochondria were once free-living prokaryotes that were efficient at aerobic respiration, and they were engulfed by a larger eukaryote, and instead of it breaking it down with the lysosome, it formed a relationship, a symbiotic relationship. And remember, the evidence for that is the double membrane that's around it, and also the fact that it has its own circular DNA and ribosomes. And there's kind of a cartoon showing you the whole thing. All right, I hope that helped. If you have any questions, let me know.